Hello, everyone, and welcome to today's live broadcast, Application of a Novel Cell Proliferation Biomarker in Cancer Drug Development, Immunoassays of Thymidine Kinase 1 in In-Vitro and In-Vivo Studies of Hematological and Solid Malignancies, presented by Martin Shaw. He is a Business Development Manager at AroCell AB. I am Marjorie Torres of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is a leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask the Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom or center of your screen, or use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Martin Shaw. I will now turn the presentation over to him.
Sharing your time with me. The presentation I'm going to make now, everything is in the public domain, but since it is a commercial company, I have to make a statement regarding financial issues. And that is that this presentation may include predictions, estimates, or other information that might be considered forward-looking. While these forward-looking statements represent our current judgment, on future trends, they are subject to risk and uncertainties which could cause actual results to differ materially. You are cautioned not to place undue reliance on these forward-looking statements which reflect our opinions only as far of this date of this presentation. So, having got the legal out of the way, we can now get to the interesting bit. I'm going to present on improved studies of malignancies with a novel proliferation biomarker, Thymidine Kinase 1. It's going to be quite a long presentation, so I'm going to break it up into sections. First of all, I'll talk a little bit about what are tumor biomarkers, then on Thymidine Kinase 1 as a tumor biomarker, then its application to hematological malignancies, then solid tumors, then in vitro drug studies, then on its assay, and then summary and discussion. As I leave each part, I will be making a slight repetition of the key points, so I'm not forgotten myself when I repeat things. It's quite intentional. So first of all, what are tumor biomarkers? And then I'm going to talk a little about what are in particular are proliferation biomarkers. So tumor biomarkers, any substance that is associated with the presence of a tumor is a tumor biomarker. And most serum tumor biomarkers indicate tumor mass, such as example CEA, PSA, CA15-3, beta-2 microglobulin, and many more. But there are very few serum biomarkers of cell proliferation. And our hypothesis is that, and why we're interested in TK1, is that most current tumor biomarkers measure what has happened, the past rate of change, such as doubling time, or the current situation, such as tumor mass or white blood cell numbers. But what really matters is tumor growth. And proliferation biomarkers are important in measuring tumor growth, as I mentioned, and you can use this information to stratify patients and subjects. You can use it to aid a choice of therapy. For example, if the tumor has got a low proliferation index, maybe you should watch for waiting, or use less aggressive therapy. And if it's high, then you should use more aggressive therapy. And furthermore, many cancer drugs are toxic or have side effects. They're expensive, and there's a chance they would drive tumor resistance, so you don't want to use them unnecessarily. The proliferation biomarkers that are available today are mostly immunohistological biomarkers, and the gold standard is called Ki-67, and there's also PCNA, and the one I'm going to talk about most for the rest of the presentation, TK1. These are all proteins that are expressed during the growth phase of cells. And the percentage of cells which stain for these biomarkers is the proliferation index and provides an indication of the current cell growth. But there's certain problems with them. First of all, they require biopsy, so they're invasive, and they only show the status of a small part of the tumor, and they have a low reproducibility because they're opinions. So there is a need for a biomarker that indicates the total body activity of the malignant disease, that is applicable to a standard serum sample, that can be determined with a routine analytical method. And we're going to propose that the serum thymidine kinase 1 measured using an antibody against the TK210 antigen has the potential to be such a biomarker. Talking about what TK1 is, serum TK1 enzyme activity has been used as an established proliferation biomarker, mainly for hematological malignancies for about 35 years. It's been studied in many types of solid tumor, which I'll come to later on in the presentation. And when TK1 is used in immunohistological staining, it is highly associated with the gold standard proliferation biomarker Ki67. What does it do? Thymidine kinase 1 is involved in DNA synthesis. It couples phosphate to thymidine to make thymidine monophosphate, which is a key step in DNA production. And this is the reaction which is involved in the commercial assays for TK1 activity. And TK1 is released from the cells during their turnover and eventually ends up in the body fluids. So that's the introduction to the proliferation biomarker and thymidine kinase 1. Now a little bit for its application. In solid tumors, which I'm going to come back to later on, its enzyme activity in the tumors is high in most forms, not all. But now going on to the next part of the presentation, TK1 activity in hematological malignancies. Here's a summary of its elevation in different types of hematological malignancies. As you can see, it's elevated in most forms of hematological malignancy, but the extent of the elevation varies from malignancy type to malignancy type. But what you can do in the hematological malignancies, you can use it to study the disease. You can use it as a discriminator of disease stage. It can provide prognostic information. It can aid in studying therapy response and relapse and in disease progression. Looking at the first part of this, clinical staging, here's a study on non-Hodgkin's lymphoma, and as you can see, as the volume of the tumor increases, the serum activity of thymidine kinase 1 increased as well. So there's a good agreement. Looking at chronic lymphocytic leukemia, another form of hematologic malignancy, this is the association of the TK1 enzyme activity with the disease stage, but using the RAI staging system. And you can see that as the stage increases, the level of TK1 in the serum increases. And interestingly, the solid circles are cases where the disease is a very slow course, so-called indolent disease. And there you can see that it's solid. The squares are ones which were initially thought to be indolent, but then later progressed. And you can see that these are somewhat higher than the ones which stay indolent. So it seems to be prognostic as well. Looking at making prognosis, here you have a study on chronic lymphocytic leukemia. And there you can see that the ones which have a low level of TK1, that's below 7, have 50% live for about 7 years. Whereas the ones that have a high level, 50% live only for about 18 months. So it's a very strong prognostic indicator. Here you have similar in non-Hodgkin's lymphoma. And here again, the ones which have a lower level, they have, again, a 50% chance of living about 7 years, whereas the ones which have a higher level have a 50% chance of living 18 months or so. So again, it's a good prognostic indicator. Looking at disease progression, here is a study on acute myelolytic leukemia. And here is the change in TK1 serum activity in subjects with different outcomes. The top two are subjects which survived during the period of the study, and you can see that the TK1 level falls after therapy and stays low. The bottom two are for subjects who died, and here you can see that there's a rapid increase in TK1 activity prior to death. So it does seem to be a monitor of disease progression. An important part of biomarkers is that they're combined. Different biomarkers show different things, and the combinations usually provides extra value. Combining serum TK1 with other biomarkers, it seems in some cases that the serum TK1 activities reflect a combination of tumor mass and proliferation rate. You get some idea of the total number of cells growing, I should say. It can also, the serum TK1 value correlates with the proliferation index, which is the measure of cell growth. And it also correlates with the peripheral blood lymphocyte count to give some idea of the tumor burden. So it's a very useful biomarker in studying the therapy response, tumor growth, can be used well, combined profitably with other biomarkers. So why has it not been widely used? And the state of TK1 in serum has got much to do with this, because intracellular active TK1 is a tetramer. But serum TK1 exists as complexes of different sizes and enzyme activities. And some sera contain TK1 inhibitors, and serum enzyme activity assays may underestimate serum TK1. Then the gold standard assays for TK1 utilize radioactive traces such as radioactive tritium or iodine. Commercial assays may require specialized equipment, but immunoassays can solve these problems. So now I'm going to very briefly talk about the immunoassay of thymidine kinase 1, and I'll come back to this again more fully at the end of the presentation. As I mentioned, TK1 in serum comprises complexes of different sizes and different enzyme activities, but they all express the TK210 antigen. Here is a FPLC study of enzyme activity against molecular weight in a fraction of serum. And as you can see, there's two peaks, one about 720 kilodaltons and one at somewhat around 100. And these fractions have different enzyme activities, but they all express the TK210 antigen, as can be shown by Western blotting. The assay itself is a simple microtiter plate sandwich ELISA. It utilizes monoclonal antibodies against a unique amino acid sequence on the TK1 monomers, the TK210 antigen. And importantly, it's a unique pre-assay treatment releases TK1 from the serum TK1 complexes and exposes the TK210 antigenic sites. It gives a very good correlation with the gold standard radioassays of TK1 activity. And looking at the diagnostic power of TK210 ELISA compared with the TK1 activity assays in leukemias, you see that the diagnostic power is very similar. The area under the curve for the TK210 concentration is 0.84, and for the TK1 activity, it's 0.79, with very similar sensitivity and specificity. So this means that you can relate results with the immunoassay to the traditional historical data using TK1 enzyme activity assays. Looking forward, in studies in hematology, here's a very preliminary study on TK1, TK210 serum concentrations of protein. You can see that in subjects or controls, the ones with the green, and then when you have subjects pre-treatment, it's somewhat higher. And then during treatment, the results are more variable, generally a bit higher. And then in subjects in remission, it returns to close to normal. So it does seem that we have got proof of principle that this assay can be used in hematological malignancies, and that the data can be compared with the historical methods based on enzyme activity assays. So looking at hematological malignancies, TK1 is a biomarker for cell proliferation, difficult word to say. Serum levels reflect disease activity. TK1 can aid in patient or subject classification, can be used to make prognosis, can be used to monitor disease progression, adds information to studies performed with other biomarkers, and can be used in many malignancies. It's a standard microtiter ELISA, gives a good agreement with clinical results based on TK1 enzyme activity. It's an open system. and it's CE marked. I'll now go on to another field, which is TK1 in the study of solid tumors. And we can discuss your comments on hematological malignancies at the end. As I mentioned previously, TK1 is elevated in solid tumors, most of them. And here is a selection. And it also seems to be that the expression of TK1 in these tumors is related to therapy response. For example, a high tumor TK1 activity was a marker of poor clinical response to tamoxifen therapy. So it is possible that TK1 can aid in therapy selection in solid tumors. A nice research idea. So why has TK1 not been widely studied in solid tumors? Back to this one again, you've got this TK1 is present in serum in stable aggregates of many molecular weights and enzyme activities. As I mentioned, they all express the TK210 antigen. But many of the TK1 aggregates in serum from solid tumor subjects are enzymatically inactive, although they express the TK210 antigen. And here you have an example of this from a subject with prostate cancer. If you look at the enzyme activity based on the FPLC fractionation, you can see that the hollow circles are the normal, the activity in the normal subject. And then you see that the activity in the prostate cancer subject is at a somewhat lower molecular weight. But look at the bottom and look at fractions 14 onwards. You have a lot of TK1 there based on the Western blot studies, but there's no enzyme activity. A lot of the TK1 protein in solid tumors, serum from solid tumor subjects is enzymatically inactive and so is underestimated using enzyme activity assays. Repeating again, the assay is a standard microtidal plate ELISA. But to emphasize here, there is a unique pre-assay treatment that breaks up the serum TK1 complexes and exposes the TK210 antigenic sites. And you can see that this increases the diagnostic sensitivity assay. If you look at the TK210 concentration, the sensitivity is about 0.5 and the area under the curve is about 0.9. On the same subjects looking at the enzyme activity, the sensitivity is only about 0.26 and the area under the curve is only 0.79. So in these breast cancer subjects, the TK210 ELISA was about twice as sensitive for detecting the TK1 protein. I mentioned combining TK1 with other tumor biomarkers, and here's a study on breast cancer. The blue line is the CA15-3 biomarker, and then the red line is the TK210. But then, and you notice that TK210 may be somewhat more sensitive, but then if you combine them, you get a better result still. And I think this is where TK210, TK1 immunoassay is very powerful when it combines with other biomarkers. There you can aggregate there's an indication of tumor mass with an indication of tumor rate of growth. Here's another subject in prostate cancer. And here you can see the comparison with a form of PSA called Pro-PSA, which is suggested as being more accurate indicator of prostate cancer than the normal total PSA. And you can see here that the red line is the Pro-PSA. The blue line is the TK210, very similar power. And then the green line is the combination, which is better still. So it's it's very valuable to combine TK210 with other biomarkers. So summarizing the TK210 ELISA in solid tumors, the AroCell TK210 ELISA measures TK1 forms missed by enzymatic assays. The TK210 ELISA better discriminates between healthy controls and subjects with solid tumors, can be used to study many types of tumors, adds information to studies performed with other biomarkers, and it's CE marked. So that's the solid tumors, and that's a description of the use of the TK210 ELISA in vivo, and I hope I've convinced you that it's a very valuable addition to the study of malignancies. I'm now going to talk a bit about drug discovery and the potential role of thymidine kinase ELISA in this. Preclinical evaluation of novel compounds in cell culture is a key step in drug development. And the assay of TK1 with the AroCell TK210 ELISA offers the potential to study anti-proliferatory effects in vitro. Here's a study where we have lysed leukemic cells and compared the amount of TK1 released with the number of cells, and you can see there's a good correlation, I should say. Now looking at the response, and this is a bit complex, but bear with me. Here you can see the effects of the drug doxorubicin on a culture of leukemic cells. And you can see that as the concentration of doxorubicin increases from 0 to about 0.1, the intracellular TK210, TK1 concentration increases and then decreases with the higher doses. And at the very high doses, the TK1 is released into the culture supernatant. This is very similar to what is seen in patients where the administration of drugs that affect the synthesis of DNA or cause DNA damage seem to induce TK1, and this is reflected in the fact that using positron-labeled thymidine derivatives in PET scan, you can see that the tumors seem to flare with the early stage of therapy. So what we can say is that the induction of TK1 at low doses in drug culture seem to be similar to what's seen in vivo. And this means that TK1 assayed with the TK210 ELISA is a potential translational biomarker because the results that are seen in cell culture seem to be similar to that which is seen in vivo. So there you're now seeing the applications of the immunoassay of TK1 in vivo and in vitro. I'm now going to very briefly present the assay method itself. And to go through this, first of all, a little bit about the challenges of TK1, and then the procedure itself, a little bit of performance data, a bit on sample stability, a bit of further information, and then discussion. As I mentioned, the molecular weights of serum TK1 complexes and the specific activities differ between normal and tumor subjects. And some TK1 aggregates in serum from subjects with solid tumors are enzymatically inactive, but they all express the TK210 antigen. As I mentioned, in hematological malignancies, the distribution of enzyme activity and the presence of the TK210 antigen is quite similar. So you get a good correlation with the results from the enzyme activity assays. But in subjects with solid tumors, where a lot of the TK1 may be enzymatically inactive, the ELISA is more sensitive. So it's very important that the pre-treatment of the samples that breaks the TK1 tetramers from the high molecular weight complexes and exposes the TK210 antigen using the the sample diluent. And this gives greater sensitivity for solid tumors, and also it gives you more consistent results between subjects with different types of malignancies. So that's the reason why the assay is more sensitive. Now coming into some of the methodology, as I mentioned previously, it's a microtiter plate sandwich ELISA, uses monoclonal antibodies against a unique amino acid sequence, the TK210. And importantly, again, a unique pre-assay treatment releases the TK1 from the serum complexes and exposes the TK210 antigenic sites. Here you see you had the pre-treatment. You add the sample to a microtiter plate coated with anti-TK210, the green antibody. You then add a second antibody, second anti-TK210 antibody labeled with biotin, that's the red. And then you develop the color using streptavidin labeled horseradish peroxidase, a very standard microtiter ELISA. It's got a good assay range, it's sensitive, It gives linear dilution, it's very precise, and it's stable, so you can have the kit in the refrigerator waiting for samples to arrive. The samples themselves, TK1 is quite easy to work with. It's got a stability at room temperature of 24 hours, so you can handle it quite easily. It's then stable at 4 degrees centigrade, so shipping should not be a problem. And then you have good stability when frozen, so you can do retrospective studies or collect samples until you've got enough to make the assay, to make it practical to run the assay. As I mentioned, in hematological malignancies, you have good correlation with the enzymatic assays. And similarly, you have a good diagnostic correlation with the enzymatic assays in hematological malignancies. But in solid tumors, you get a much better sensitivity than compared with the traditional methods. It combines very well with other biomarkers. We are now starting to publish articles based on its assay. So to conclude a long presentation, TK1 is a biomarker for cell proliferation. Serum levels reflect disease activity, and you can use it for patient subject classification, prognosis, monitoring disease progression, can be used for many malignancies. TK1 has different forms in solid and hematological malignancies, and enzyme activity assays underestimate TK1 in solid tumors. But the AroCell TK210 ELISA measures all forms of serum TK1. There's good agreement with clinical results on TK1 enzyme activity in hematological malignancies, greater sensitivity for solid tumors, more comparable results between types of malignancies. It's a standard ELISA procedure, positive synergy with other biomarkers, applicable to in-vitro models, an open system, and it's CE marked. So, the AroCell TK210 ELISA, a novel biomarker of cell proliferation, which, as I say, measures what really matters, the rate of cell growth. It expands and simplifies the study of cellular proliferation. There's new opportunities in cancer research. And thank you for listening to me. It's been a long talk. I make the meeting open to discussion. Thank you very much for your attention. I finished.
Thank you, Martin Shaw, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your questions into the box that appears on your screen and click on the Send button. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, do you have any data on xenografts?
We have data that human TK1 can be measured in mouse serum because the mouse TK1 has got different immunogenicity to human TK1 when you measure the TK210 antigenic site. So we know that it's chemically possible, but we have no data showing whether it can be used to study xenografts in mice. If anybody's got such a study in mind, I'd be very pleased to hear from you so that we can do something together.
Thank you for that answer. Our next question is, why are there so few recent studies on TK1?
I think this is because the assays based on enzyme activity were developed a long time ago, and based on their limitations, the research went as far as it could. Now that immunoassays are being developed, I think you're going to see a new interest. And we at AroCell are now starting to publish data using the immunoassay of TK1 using the TK210 antigen. And I think there'll be many more published in the near future.
Thank you for that answer. Our last question is, I noticed that the cutoffs between better and poorer prognostic scores differ between malignancies. Any comments?
I think this is because different types of malignancy have different proliferation rates, and also they're found in different age groups. For example, chronic lymphocytic leukemia is tend to be found in very old people, and we have noticed that the age ranges for TK1 are age specific, particularly in elderly people. So I think this based on the type of patient and the aggressivity of the tumor.
I would like to once again thank Martin Shaw for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through May of 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.